Uh, we'll wait a few more minutes. We should get started. Like we say, 2 minutes. Don't worry. In France, we're also late, so don't worry. The organization for today. So we had, if I'm trying to explain how I've been trying to build the lectures of the week. So on Monday it was a general introduction about the scale, spatial scales, coming from the molecular scale where the meaningful information is standing, going through the cell, multicellular system, organ, organism. No, so the very first day, basically the first hour. And then I went through scaling arguments where I was trying to convince you that the molecular scale, which is the relevant one for the DNA, for the RNA, for the protein, and for the lipids, is embedded into a world which is non-intuitive. It's totally dominated by KT, this is Brownian motion that we discussed about, and innate matter. And then there were scaling laws that I was sharing with you, which are quite easy, I think, to reconstitute. You remember, energy scale, KT over A gives a force, piconewton forces, that you can measure with optical tweezer. Einstein relation with the diffusion constant, which was the ratio of KT over 6 pi eta A, which was basically the ratio of jiggling around over the friction. And then I went to the Reynolds number estimate to give you a hint that, in fact, this biological world is dominated by viscosity. So this was the very first day in the morning, somehow. And I'm trying to convey that it's a very counterintuitive world in the sense that jiggling around all the time at the protein, molecule, DNA, RNA level, everything is jiggling around all the time. I understood that some of you are working on disorganized domains. They are jiggling around all the time also. And somehow this is a given. that somehow the biological system have to cope with this reality, which is that jiggling all the time. And just to reconstitute the fact that it's counterintuitive is that I think one has to imagine a protein or a molecule inside the nucleus or inside the cytoplasm, which has an amazing large diffusion constant. Yeah, it's 5 micrometers squared per millisecond. So it means that if the cell is this room, a tiny bit like this would be going at the end of the room, within a millisecond. So it's the way actually the physical rules are set for the cells, which is a problem, because if you want to have convey an information that is meaningful, you'd rather be specific whenever you need a transcription factor to reach a target point in the genome. So basically there is a problem, right? So that was the morning. In the afternoon, we went to the lab to observe the motion of 3 micrometer beads particle and 6 micrometer beads particle. And for this, you use the foldscope as well as the microscope that Pramod had prepared. And you could see that the beads were jiggling around. And all of you, I think you've been acquiring some data that you shared yesterday evening on the folder. That was the day one. Day one was really inert material. That's the particle jiggling around. There is nothing special, no biology at this stage. It's a pure passive process. Day two, meaning yesterday, on Tuesday morning, I was trying to move forward to basically trying to ask the question, how do you go from this passive reality of passive rules to active rules? And I was writing on purpose active/living matter. Being vague in how do you translate an active matter into a living matter, but at least posing the problem that there is a way to move to active matter. And for this, I was stating a different list of points. Point number one is that there is an energy source that is given by ATP hydrolysis. So basically, ATP hydrolysis is going to give the energy to any biological system, from budding yeast, fission yeasts, to plants, flies, embryos, human embryos, and so on. So ATP is the source, plus other sources that I mentioned yesterday also, which are the NTP, CTP, TTP, GTP, all these bases which are composing the DNA molecule. So this is an energy source that is going to drive the system out of equilibrium. So I'm making a clear distinction between these 2 days because just to convey the message that living matter has to be competing or cooperating with this passive world, which is imposing diffusion and so on. So it's energy driven, out of equilibrium. That was another point. And the third point I was conveying yesterday was the fact that all the biological matter is piloted by molecular motors. So that was basically this point that I was starting by yesterday morning. And I went through the list of molecular motors, one after the other. It was not an exhaustive list, actually. It was just a table where I was listing out. Could you remind me the molecular motors that I was giving? Yes. Dynein. So it was always a pair. So there was a biopolymer on top of which there was a motor moving. So there was microtubule kinesin, microtubule dynein, another one? Actin and myosin. So actin was the filament and the myosins were moving on top of it. What else? Which other motors were like ATPase. Yes? ATPase. So F1 ATPase, which is a rotary motor. Nice. Fourth one?
Ribosome and mRNA.
DNA.
Absolutely. So ribosome, mRNA, which is doing protein, DNA polymerase and DNA, RNA polymerase and protein, helicase, topoisomerase, you name them basically. As soon as you have an ATP hydrolysis and a directed motion, they are eligible to be called molecular motor. So it gives an intuition which is interesting, is that as soon as you see a directionally moving molecule, which I was showing in the movie, somehow at this scale, it's a motor. So it has to be using energy, point number one. And point number two, there has to be a broken symmetry, which I was insisting upon yesterday, because it's me walking like this. So I'm a kinesin, I'm moving on my microtubule and I'm moving straight. So the reason I was insisting so much is that this table that was here on the table, on the board yesterday, all these molecular motors have essential functions. Microtubule and kinesin dynein, their function is to transport. So then it will allow specifically to transport a vesicle to fix a point that would be damaged inside the cell. Actin and myosin, the function will be contraction of the muscle. This is a perfect crystal structure. Historically, that was amazing to take a muscle, send X-ray, and then have a perfect cliche of Brad Pitt's. It's an amazing cliche, but in fact, throughout the body, you see actomyosin, which is shaping the cells, shaping the embryo, shaping the organs. So it's an ensemble of actin and myosin that are doing the job. And for RNA polymerase or DNA polymerase, well, we say? Yes, absolutely. And so it's an essential motor. And I was trying to take the example of the reading here of, let's say, global initiative on academic network course. I can read this because I'm going from the left to the right and I'm reading letters one after the other. So DNA polymerase is reading essentially one after the other. There is Brownian motion, so sometimes the DNA polymerase go backward and there is an error, but eventually there is the directed motion piloted by this energy source. So with this simple statement of directionality, you see that many of the key functions of the cells of living matter are conveyed. Reading, preserving, transcription, translation, duplication, cell division with actomyosin, transport. You need these molecular motors to be transforming an inert matter into an active / living matter. Now there was no problem. There was another point actually that I was trying to convey is that there is a broken symmetry, and this broken symmetry is essential to give a direction, right? So that there is a track. The DNA polymerase has to read from one direction and not the opposite direction. 3', 5'. There has to be a directionality in the motion. The same for actin and myosin, kinesin, microtubule. And this was the last point of the lecture of yesterday. I was trying to convey the message that this broken symmetry was carried by the chirality. Yeah, chirality. So basically, since the motor, the combination of a polymer on the one hand and a molecule on top of it, there is a broken symmetry that is encoded deeply inside the structure. And this is a given. Somehow that's the way it is. So there is a broken symmetry inside each of these biopolymer, and there is an additional broken symmetry for which we had a very, very stimulating debate yesterday, is that, okay, the biopolymer has a broken symmetry, but whenever you have the motor interacting with is this interaction also has a broken symmetry? And the short answer is yes. So it's a combination of broken symmetry of biopolymer plus the motor, which eventually leads to the directionality. And I was, the final point of yesterday was to show motility assays experiments where over the decades, many scientists across the world actually have been taking these molecular motors out of the cells and reconstituting in vitro the motion. It's a so-called motility assays experiment. Wherever you have a flat surface, on the surface you graft the motor protein and then you put a filament on top of them and you see them moving like worms. I think many of you were saying, oh, that's like worms. These are biopolymer, right? So they're not living entity, it's biopolymer, but they have a directed motion, so they move like this in a directional manner. And the point I was also trying to share is that the motion to be directional, the broken symmetry of the biopolymer is essential because it's coming to the surface, it's going to interact with the motor, and somehow it is the chirality, local chirality, that is going to allow the myosin motor to attach and then give a kick in the right direction. So far, so good? Okay, good. So that was yesterday morning, and then we had debates, when does the active matter start, when does it stop, when does living matter start, when does it stop, okay, it's not living material, is a directionality a proof of being living, this is more epistemology than raw science, but it's a very interesting question also, is that when does it start, what is the frontier between this inert matter and active matter. But the punchline is really like energy source and out of equilibrium is at the core of the transformation from inert matter to living matter. What is the order of magnitude of ATP hydrolysis? 10 KT. So if you could remember one thing of yesterday, first day if you could remember KT, and second day if you could remember 10 KT, you're all set. Essentially, the fact of thinking about 10 KT provides the energy to outcompete KT. Orders of magnitude, right? You look for an energy source. KT is the energy that is jingling around all the time. So if you want to uncompete, you need to imagine a system that would be 10 orders, or 1 order of magnitude larger, so that eventually you get a motion that can compete. In the afternoon, we went back to the lab, and you used yesterday, you had amazing movies. I mean, I've been, browsing to what you've been doing. I think, yeah, thank you very much to Pramod again, because I think, and so, what did you see in the movie yesterday? This was pond water. Pond water from campus. Both places were... Yes, and? Okay, so, yeah. These were the two places. What did you see under the scope? Microorganism. What was the motion like? Directional, yeah. I mean, the first point, I mean, the first 10 minutes when you started having the observation, I could see on your cell phone with the small microscopes, with the foldscope, which is amazing quality. I mean, this is the foldscopes are really sensational. You could see motion, but what type of motion did you see? Directional, fair enough, but was it identical or it was different? What types of diversity of motion did you see? Diversity of motion. Could you do it with your hands, typically? Yeah, what else? Yeah, what else? Yeah, rotation. Clockwise, counterclockwise? You are in opposite direction. Yeah. Okay. The size was the same? Velocity was the same? No, okay. So you had a diversity of directional motion, right? Rotation, translation. I was not in New Delhi before I arrived, so I discovered these motions together with you, right? So basically, it was... and you had all kinds of microorganisms that you observed. I think one of you was making the hypothesis that this was a predator-prey system. I don't know if he's in the audience now, but basically, it's a world. The pond water, they need food, so they eat each other, right? And so probably in one of the systems, it was really a predator-prey system. So basically, you saw yesterday that directed motion is existing, and bear in mind that this is at low Reynolds number, lecture number 1, right? So they are moving not like you swimming in a swimming pool, where in fact, if you stop applying a force, you continue to move because there is inertia. They have to apply at every single point a new force. Every time point. Right? They have to apply a force. And another point that I will not dwell on too much now, but I will come back to it at the end of the lecture of today, is that in addition to basically applying a force at every single point, because force is compensating by velocity and not by acceleration, they have to have also a broken symmetry. This is not an intuitive idea that was popularized by somebody named Purcell. There's an article I think we'll study on Friday, which is Life at Low Reynolds Number, is that if they move, they are using ATP hydrolysis. All of them that you saw yesterday, all of them are using ATP hydrolysis, all of them. All of them having molecular motor. And the idea of the lecture of this morning is to try to introduce you of how do you go from single motor to an ensemble of motor. That would be basically the game of the lecture of this morning. So the menu for today is 2 hours lecture about, if you wish, collective effect of many motor interacting together. How can you design them? What are the building blocks? How can you write equations to predict how they're going to behave? What is the problem? Why it's not an easy problem? Why it's not simply intuitive? And this afternoon, we'll analyze your movies of day 1 and day 2. So we'll be in this room this afternoon. Everything is on the Google Drive, so we'll have access to all the data of everyone. And the game will be to extract out the tracking of the first day, diffusion of the particle, second day, the rotation or the jiggling around, and to characterize them with ImageJ, with TrackMate, and then to extract number out of it, to characterize them, with numbers. And I'll give a research talk, which is more what we do in my lab at 5:15. That will be an example now of research that we are doing with a comparison between experiment and theory. So the hardcore, if you wish, comparison between experiment and theory would be rather at 5:15 because in one day it's impossible to, I think, at least to me, to go from the experiment analysis and then comparison. So the comparison, strict one, would be rather in the research talk. So far, so good. Any questions or comments? Good? Right. So let's go. The more you interact with me, the better. My impression is that you prefer chalk than PPT. Am I right? Okay, good. That's I also prefer chalk. So basically, I try to minimize the PPT. I need to show you some images sometime, but I'll go through the chalk. And again, you're welcome to interrupt me anytime. So the topic number 3 is going to be tightly related to basically cell motility. How does the cell move? How does the cell crawl? Yesterday they were swimming, all of them, right? And the question is that what are the building blocks that are allowing all these systems to move, to crawl, or to swim? For this, I need to pose the problem, right? To say, well, there is a problem, and then build things one after the other. So I insisted yesterday, and I repeated that this morning, that there is a need for a directionality. Right. So I'm going to depict the biopolymer as an arrow. This can be any biopolymer. I don't care, DNA, actin filament, microtubule. And I'm going to take another color for the motor. It can be transporting a cargo, it can be doing something else. Let's imagine that it is interacting somehow in this direction, and then I have another motor that is like this. You're going to see that I'm starting to have a problem as soon as I'm going from one filament to two filaments. Let's imagine that I have this configuration. And let's compare this situation with that situation. What's going to happen in this case? What's going to happen in that case? Yes, you are making gesture, very good, but you are making gesture in opposite direction. So you need to tell me, let's call that A, and let's call that B. What's happening in A, what's happening in B? I would like the women to start. Priority to women. What do you say? This one could be very fast. Why?
Because there's only
one direction. Okay. So velocity, I don't know, but I think I see your point. Why do you think that this one is going to be more efficient? What is your? What do you say? Less friction. So basically, where is it going to go? The two motors here are going to go where? Yes. Yes, to the right. So basically here, you expect that later on, they're going to accumulate here. That's correct? Okay. What's going to happen to these ones? Yes? It won't move. I mean, you have the impression that if there is some bound here, there is a kind of contradiction between here, the molecular motor moving to the right, and here, the molecular motor moving to the left. It could stretch. Now, I need to imagine that there is here a wall to prevent it from moving. But you see that already with two filaments, there is a problem. And it's not a minor one. It's a major one. Because here, I can end up with basically a transport like this. Here, I can end up with a fourth generation, which is totally the opposite, right? If I'm taking here, and I hope I won't break anything, but basically, oops, I broke something. But you see what I'm trying to say. Either I'm moving it, or it is attached and somehow I'm going to apply a stress. It is very different mode of action on the matter. So with this very simple example, already started at 2, which echoing the discussion we had yesterday afternoon actually, or at the end of the morning, only by playing with polarity and with molecular motor, immediately there is a problem. It's not like a detail or an epiphenomenon. It is at the core of the problem. Everybody is with me because it's going to become more and more complicated. It's okay? So whenever you go from 1 to 2, you get opposite results. So polarity, I mean, there are different ways to call it. I was calling it chirality, but I will use the word a lot, polarity. Basically, there is a front and the back. The fact that the biopolymer has a polarity is going to generate, if you wish, a richness or a problem. So the fact that it's already, it is already a problem when I have 2, you can imagine now if I have 3, 4, 5, 10, 10 to the 2, 10 to the 3, 10 to the 4. It's becoming a very difficult problem because if I'm trying to mix up all the polarities, I can try to draw as many schemes as I want. I would be incompetent to be able to predict anything. Because here, this is simple, but let's try to play the game. I will simplify the scheme. Imagine I'm drawing this. I'm simplifying my life by drawing only the polarity like this. And I'm having now this. I'm doing 4. And now I'm repeating the game of putting motor like this. In which direction this is going to move? What?
More or less it stays in the same place.
More or less it stays on the same place. Any other guess? Yes. It's really to the right. Why? Because there is one more in this direction. Any other guess from anyone? Who says it's staying at the same place? Same place, yes. Why? All the same place. Who says going to the right? Who's saying is going to the left? No one. Who has no opinion? So basically, you see that here already at the problem, there is already I did only 2 more, and now I'm incompetent to be saying something fair. And if you go at the level of a cell, which is the level we're going to deal with actually today, essentially, it's not 4 that I have. It can be locally, I can have 10 to the 4 filaments. And then there is no way I can predict anything. No way. Then I need to compute equation. I need to write it, because then I'm entering into another reality, which is the reality of collective effect. I have many filaments, many motors that are acting in concert, and then you need to write equation, compute the equation, compute the result, and then compare the experiment with the theory. So I want to start by this because the rest is not very interesting if the problem is not striking. You see what I mean? That there has to be a problem first. So yes?
Is it something like crowding effect?
It is a mixture of crowding effect, but crowding effect, to my knowledge, sometimes can be associated with, if you wish, a kind of high viscosity. So basically, if there is highly viscous, motion will be impossible because there is an effective viscosity that prevents the motion. Here, there are 2 effects. This is, it can be in low or high viscosity, but there is, if you wish, a tug of war between an assembly of molecular motors interacting in concert. And for this, as I will try to show to you today with simple example, you have to know the polarities, you have to write the equation of interaction of these objects. Only the results of the equation can give something meaningful. You see what I mean? Because I can try to hand wave the system, I'll be blocked at some point, because you see the intuition goes in opposite direction, and each of these intuitions are equally correct.
Is it possible that net is a 0 effect?
It could, but it could maybe not. That's the whole point, is that if you tune the activity of the motor in the right way, if it is localized in the right way, if you tune the force velocity of each of these motors, you might end up with a directed motion. So that's why intuition is basically very misleading here. So that's why if you wish the collective effect, which is, if you wish, the fact of it's the tramway metaphor I was taking the very first day of an ensemble of individuals inside the tram. One of you told me he was working on big check model. Where are you? Basically, toner 2, big check model. Yes, right. So basically, it's typically a collective effect. Whenever you have many particles interacting in concert, you will have a new effect. This is what I'm talking about here, together with the molecular motor action. So if the problem is fine, basically, you will have this problem for most of the molecular motors I talked about yesterday. Yesterday, I was restricting myself essentially to the interaction of a single motor with a single filament. It's happening inside the cell, but it's not that common, right? You can have isolated this type of event. Kinesin transporting cargo inside microtubule, this will be typically this type of situation. But most of the time it will be collective effect, and this is what I'm going to talk about now. Okay? It's okay?
Yeah.
Good. So now I'm going to another point, which is going to be the presentation of the cytoskeleton. Who knows the cytoskeleton? Maybe many of you know it. For those of you, basically, could you remind me the three main components of the cytoskeleton? And we need them today, obviously, to go to cell motion. That's right. So you have actin filament, intermediate filament, and microtubule. And then we need these three, actually.
So there is an online question here.
Yes.
So they are asking what is the effect on global cellular movement with increasing polarity?
If there is an increase in polarity, how is it? This is this is the core of the lecture of today until 7 p.m. So I hope by the end of the day I'll answer.
Okay.
So we have some audience online also joining.
Sure, with pleasure. But may I delay the answer?
It's the online lecture maybe.
Okay, sure. The more you interrupt, the better, right? It's the whole point, right? And so you have basically these three structures. And for those of you who are not familiar with these structures, I need to show them on the scheme because I think that will be better. At least I give you the dimension. So this is 7 nanometer in diameter. This is 23 nanometer in diameter. And this is typically 11 nanometer in diameter. So these three structures, actually, so it's 7, 11, and 23. The fact that it is called intermediate filament was historical, essentially, because they have a diameter. It was discovered in the beginning of the 20th century. The diameter is in between actin and microtubule. The three are essentially chiral structures. This, I think I've been over-repeating it, but I'm repeating one more time. And I'll show you now a scheme to convince you that they are indeed chiral. So this is taken from a textbook that I invite you to read. For those of you, some of you are starting or learning biology actually with other backgrounds. So Molecular Biology of the Cell is a very good book. You probably know it, most of you. It gives the basics very clearly. So I'm showing you the scheme of these three molecular motors and cytoskeleton structure, and you will see immediately actually the structures appearing. Yeah. So now you should see the structure appearing in a second. So you see the actin filament at the top. This is an old picture. Nowadays, with cryo-electron microscopy, one does much better. Actually, you can even reveal the polarity with no marker with cryo-EM. So one of my colleagues at my institute, Florian Fessler, has been revealing polarity with this in cells. You see at the top what the scheme look like. You see at the bottom, right here, you see the actin filament. It's really a double helix. I think it seems surprising for some of you because we are all used to DNA being a double helix, much less for actin filament. But you see that, well, you have the pitch, you have the helical pitch. So the structure has an intrinsic broken symmetry. It was the polarity I was mentioning to you before. Microtubules are here. So you see the diameter is much larger. You cannot guess, I mean, the image quality is not that good here, but you can see here that it's composed of a dimer of tubulin alpha and beta, and you see the black and the white here, which are making a helical pitch again. So there is a broken symmetry also in this structure that you can see also in the microtubule. It will become clearer in another scheme that I will show later on. Intermediate filaments are achiral, so that's the exception. I won't talk about intermediate filaments. Somehow they are very important. There are diseases associated with defects in intermediate filaments, but most of the presentation of today for moving cells, crawling cell, or swimming cell will be essentially related to phenomenon of actin filament and microtubule. So far so good? Yes?
Where do you find the intermediate filaments?
Almost everywhere. In every cell. Plants are more complicated, but in mammalian cells, you find keratin, which going to from the air, vimentin, you name it. Basically, these proteins are almost in every cell. Back on the envelope, they've been associated historically to the mechanical integrity of the cell. But if you wish, they are the poorly treated structures because they don't have motors associated to them. They are achiral. So most of the research has been focusing on actin and microtubule, but they are key. Again, there are some diseases associated with their defects. Yes.
So they give a stiffness.
They give a stiffness. Back on the envelope, the key message that people are taking is that they give the stiffness.
Would spectrum be an example?
What do you say?
Would spectrum be an example for intermediate?
I would need to double check if they are eligible for it. And this we'll talk. I'll double check to be absolutely sure. I'll need to double check if they enter. The definition is extremely strict somehow, so I need to be cautious on this. So basically, this is the structure, of each of this protein. Now I'm going to. I think it's better that I return to the chalk. So this was just the image that you had it right? And now I'm going to remove this. I will draw from time to time. I return to it, but I think it will be much clearer. I'm going to start now by describing the actin. I have two options. Either I start by the microtubule dynamic or by the actin dynamics. I think the simplest is going to be the microtubule dynamics, and then I will redo again the same cycle but with the actin dynamic. So microtubule dynamic. All the structures that I described, actin filament or microtubule, which would be our two main polymers, they have an intrinsic dynamic. So basically, the name of the cytoskeleton is not very well taken, because when you think about cell skeleton, you think about what? Skeleton. You think about what skeleton?
Some structural basis.
Yeah, so we have a skeleton, right? It's not very dynamic during our entire life. Well, maybe in the first ages, but then, doesn't change much. So basically, it's extremely static. So the word cytoskeleton is misleading. And that's parenthesis. Whenever you'll be discovering a new phenomenon in your PhD, select carefully the name of your discovery, because then 100 years after, people will use this name, and it can be misleading. So skeleton is wrong. It's good because it's going to impose a shape, but it's a dynamic. So constantly, these proteins are in constant recycling. They depolymerize, they repolymerize, and the dynamic is extremely rich. So the dynamics of microtubule is called the dynamic instability. I will depict it so that you see what I'm talking about, and I will try to make it clear. So the individual entity is called, it's a dimer of tubulin. Alpha and beta. I'm going to depict beta like this, and alpha is like this. The entity beta is capable of binding GTP, which is one of the sources of energy I was mentioning to you yesterday, and it can hydrolyze eventually at some moment GTP into GDP plus P, which is going to be a source of energy of which order of magnitude? 10 kT. So the motor is back. Right? So I'm going to have this, and eventually I'm going to polymerize and give an entity that is going to be, at the next time point, something like this. So my drawing will not be very good, and I hope you will apologize if it's not as good as it could be. But basically, I'm polymerizing the tubulin into a so-called protofilament. So basically, I'm going to have a structure like this. But at some point, if I was here in the state of GTP, and here in the state of GTP, and here GTP, at some moment, I'm going to have the structure that is going to become GTP going to GDP. And when it's coming to GDP, the structure becomes unstable, meaning that I'm starting to have a rod that is fragile and that is about to depolymerize. So then the structure is going to become something interesting. It's going to go like the structure like this, and now with only 2 dimer, and eventually if they become again GDP, GDP, I'm back to the situation, which eventually is going to allow me to return to this cycle. So you see, by doing this, I've been drawing a very simple cycle of dimer that eventually elongating, that eventually polymerize and then depolymerize. This is for a single structure, so-called protofilament. Now, if I'm going to a phase actually where I'm going to have protofilament is essentially, yes, it's okay. If I'm going to this, to now another phase where I have one end on which I'm going to allow protofilament to grow. So each of these structure is going to be depicting the microtubules that are here of 23 nanometer in diameter. And I'm allowing the protofilament to grow to form this structure of 13 protofilaments, or 15 protofilaments. Each of these rods here correspond to this line of GTP, GDP bound tubulin. Yes?
If the strand is not about to grow,
yes.
What will hydrolyze the terminal GTP to GDP?
It's spontaneous hydrolysis. In the absence of other protein, there is a time scale after which eventually there's going to be hydrolysis and then it will become unstable. So if I'm allowing this growth to have process and to operate starting a given ground here, and I'm going to allow this structure to grow, and I'm going to make my life a little bit easier by showing the helical pitch that I was talking about before, which are essentially the alpha structure or the beta structure. Everything is fine so far. If I have at the very tip here, on average, GTP bound tubulin. Because I have a cap somehow. I have a structure out of which the tubulin were growing, they grow, and at the end I have a cap which stabilizes the structure. As soon as the GTP bound end is going to become GDP, there's going to be an event where eventually, this is the basis out of which everything is growing, there's going to be a structure that is going to fade away and the tubulin will eventually depolymerize to give back the dimer. So again, the transformation of the GTP cap into a GDP cap make the structure disappear. So I have a dynamic structure. We are far from a cytoskeleton, skeleton in the proper sense. So interestingly, the community and the selection of words is very important. That's another example, actually. The growth process has been called rescue, and this step has been called the catastrophe. I'm not sure about the spelling of it, but that's okay. The spelling is extremely connotated, right? Why should this step should be called a catastrophe? Why should this step could be called a rescue? Well, maybe it's good for the cell to, at some moment, depolymerize structures. Sometimes it's good for the cell to repolymerize, but there is no fundamental reason for which it should be called rescue or catastrophe. But that's how it is. It's okay? So the molecular reaction is GTP hydrolysis to GDP, protofilaments. I have 15 protofilaments which are assembled into a rod. The rod eventually grows. It is stabilized when it is GTP capped. When GTP is transformed into GDP, it depolymerizes and eventually goes to a cycle like this. Can somebody try to guess what is it good for inside the cell to have such a machinery? What is the, So those of you who know, just at the moment, don't speak up. But for the others, try to think about why is it good for? This is the microtubule, right? They have this dynamic called the dynamic instability, growth, depolymerization. If you have some end, it starts to grow directionally, it is stable, and then it's going to be unstabilized. What is it good for? Why should a cell need that? Mammalian cells.
When cell divides, it needs to be restructured.
Absolutely, yeah. It can be a way to what? What do you gain with this?
Change the shape according to some function.
Yeah, change the shape. You can imagine that if you don't have only one microtubule, then imagine that you have 10 or 100 microtubules. And then you see that I'm going back to collective effect, right? If I don't have one now, but if I have 10, 20, or 100 microtubule, you see that eventually I can start doing something new. Change the shape, polarize the cell, and the question online about the polarity is related to what we're talking about now, is that if you have an ensemble of microtubule, maybe I'll be able to change the shape, right? So if there is no clue at the moment, I'll try to move forward with another step, is that each time there is such a phenomena, when you move to collective effect, there is always the question associated to it, is that what is it good for the cell? Does it serve any purpose? So I'm going to introduce another part in this chapter, microtubule dynamics, which is going to be one word about the centrosome. So here it was the molecular structure growing, giving a protofilament. Here it was an ensemble of protofilament giving microtubule. Now I'm going to be inside the cell. And I'm going to draw a so-called centrosome out of which project or grow microtubule. And the question that I'm asking you, for those of you who never saw the centrosome so far, according to you, why was this little organelle in the center called the centrosome? What do you hear in the word centrosome? Center, right. So the centrosome is a 500 nanometer structure. The scale of the cell is about, as I said many times, 10 or 20 micrometer. So we're talking about a structure that is 500 nanometer typically in size, and overall cell structure, which is about 10 micrometer. And the point that I would like to probe with you is that the microtubular structure plus the centrosome is allowing the cell to find its center. And for this, I'm going to try to ask you the question to get a sense of why this is the case. I need to add one more hint so that you guess what I'm going to try to demonstrate to you. The centrosome corresponds to this wall out of which the tubulin was grown. So you imagine this structure being here, and then the microtubule capable of growing like this, eventually shrinking, growing, shrinking, growing, shrinking. So the only thing that I'm giving you at the moment is, a kind of a game. The wall of this room would be the cell. This would be the centrosome to scale, right? And then you have the ability to grow microtubule out of the centrosome everywhere. Can somebody try to continue the reasoning to eventually get to this? There is a game in France, I don't know if you have it in India, which is called the Mikado. Mikado, you have rods, you throw the rods on the floor, and then you need to take out all the rods without moving the other. You said yes, so what's the name in India? What is the name of the game? It must have a name, but you see what I'm talking about. Yeah, it's a game in which you have rods everywhere. You are throwing the rod, and then you remove a rod, and so you shouldn't move. I think it's international, so yeah, okay, good. So imagine I'm giving you these rods, right? And this is the centrosome, or even better, I'm the centrosome, right? And so I have with me hundreds of rods like this. What's going to happen? I'm throwing one rod on my right, and then I hit the wall. What's happening when I hit the wall? Yes, I see the gesture, but yes, I'm pushed, right? On the wall, yes. So then what's happening to me? Yeah, I'm pushed like this, right? But I have 100 rods around me, right? What do you say? I may rotate, it's possible, yes. But you are already quick in the reasoning, but I'm starting by one, right? I'm drawing a wall here, so I'm going to be pushed like this. What's happening here on my left? The same, right? So basically I'm growing a rod here, and already here you feel that what's going to happen on my right and on my left? I'm in the center in effect. But then behind me, I'm going also to grow a rod. What's going to happen to me? I'm pushed around, and if it is behind me, eventually I'm going to go here. But I have another rod that is here, right? So basically I'm having a rod that is going to the end of the room. Where am I going to stabilize? In the center, close to you. So I'm moving like this, and I'm doing this game everywhere, so I have hundreds of microtubules which are everywhere. So you get a sense of what's going on here. So basically, the point I'm trying to defend is that whenever there is a dynamic process of the microtubule, it's not an epiphenomenon here. It seems to be contributing to this potential, dynamics where the rods are growing, they are hitting the walls, and basically my position would be a force balance between what's happening on the right and what's happening on the left. And eventually I will get stabilized at one point, which eventually will be the center. This is extraordinary, right? Because it somehow means that a little organelle can, if you wish, inform the cell about the center. And this is spanning a large order of magnitude. I was probably wrong in the estimate of the centrosome. It's about this order of magnitude, but you see here, it's like a gigantic gap of the way it can proceed. Yesterday, I think there was one of you who was asking the question, okay, in the first lecture, I was saying, how can you change scale from nanometer to micrometer through self-organization? This is an example of self-organization. You have hundreds of microtubules, 10 to 100, which eventually lead to the centering of the centrosome. So far, so good? Yes?
Can the force generated from the microtubule push the cell wall or not?
It can. It may. There has to be the contribution of the cortex, which I'm at the moment neglecting for the sake of simplicity, but there are situations where eventually it can. Most of the time, the microtubule will buckle up because of the cortex. But in principle, it might contribute. To go along your line, and this will answer the point about the polarity. So I'm summarizing where I said, find the center. If I have centrosome, if I have microtubule with their dynamic instability, eventually, this is allowing the cell to have information about the center. Now, I'm adding one more step, is that there are some circumstances where the cell needs to be polarized. Of course, when? Division. Okay. What else? In which circumstance the cell needs to be polarized? Movement. Yeah. Movement. Basically, before starting to move, before any motion, the cell is going to get a polarity. And maybe after that, it will eventually undergo a motion with a given velocity. So basically, the polarity needs to be acquired first. So I'm simplifying it to the extreme, but one way to understand the polarity of a cell is to imagine that this circular cell is going to get transformed into this structure. This is, again, another simplification, but I think for the sake of clarity, this is what I would like to show here. I have the centrosome that eventually is going to be here, and I will have longer microtubule in one side of the cell compared to the other. And basically in this stage here, if you wish, there has been, so this is not, I will exaggerate it a bit to make it more clear, but basically there can be a mechanism by which there is an accumulation of complex of proteins here which are going to stabilize at the wall the microtubule to allow their further growth. So I won't be able to do it in this room, obviously, because I cannot move the walls, right? But imagine if I would have the microtubules everywhere, pushing everywhere, and at some point, the microtubules are getting stabilized and allowed to continue to grow and push the wall, if you wish, right? Then what will happen spontaneously is that I will be moving backward, and concomitantly here, I will start to depolymerize. That's basically giving this type of structure when eventually I will have a front and I will have a back. And as soon as I do that, I'm starting to have a cell that is polarized. I haven't moved yet, but at least I am polarized. So if I'm summarizing the steps, actually, dynamic instability for the protofilament, giving the dynamics of the microtubule assembled inside the cell are going to allow the centrosome to help the cell to find the center. And eventually, if I have a cap that is stabilizing one end, I can eventually get to polarize cell. This was all hand-wavy, in the sense that, I'm pretending I'm the centrosome, I'm using games from international community, etc. But if you wish, you see, and returning to the introduction here, I can wave my hands, but it's becoming very limited at some point because you need to trust me somehow in what I'm saying, and can be a flow. I mean, it can be wrong. There can be some places where it's right or where it's wrong. So I understand that you are not many in the audience to have a math and physical background. Am I right or not? How many are biologists? Chemists? Yes. Physicists? Okay. So I will have 10 minutes of equation, not more, but just I will try to make them as clear as possible. The idea is not to impress, but just to say that at some point, one needs to delve into the equation because the brain is too limited to basically understand this phenomenon. It's helping, but at some moment you need to... And after that, you cooperate with a physicist or a mathematician who guides you in the process. But this is a step where collective effect needs math. And again, hand-wavy is good, but not sufficient. So the math that I want to show you is one model that was developed for microtubules. There were others that were developed for the actin filament. I'll say one word about it later on, but I went first to describe the microtubule. So whenever you see a microtubule, it has two types of dynamics. There is the rescue and the catastrophe. So the system has a constant growth process, eventually followed by a catastrophe where it's moving backward. It's the metaphor of me, if you wish, in the centrosome and pushing and pulling on the wall. So this, you can place it in equation, and I'm going to show the equation. They were posed by, it was a physical review later paper a long time ago by two scientists, Marilyn Doctorum and Stanislas Liebler. I'm taking one example of many in the literature, actually, but I think this one is quite pleasant to present because I think it can be quite intuitive in the way it's working. So what they proposed, actually, was to have a surface out of which you have some rods that are going to grow or eventually shrink. And you need to define a parameter, something or a set of parameters that are going to make you understand how the dynamics of the system proceed. And for this, they defined 4 parameters that I'm going to give you, and define them: F plus minus, F minus plus, V plus, V minus. So imagine you see a phenomenon and you want to put numbers on it, any phenomenon. So I will try to make you a guess, actually, what are these 4 parameters. Here I have a microtubule. It is growing and it is shrinking. I'm proposing to you that these 4 parameters, or they propose actually that these 4 parameters do the job of capturing these dynamics. Could you help me? V is what? Yes? V is the velocity. So this is the velocity of positive, meaning rescue. V is the velocity of catastrophe. F plus minus is? No, it's not the force. I have a structure that is growing, shrinking, growing, shrinking. So basically, yes? Not friction. Probability. Probability of what?
Plus to minus.
Yes, plus to minus or minus plus. I have a stochastic process. So I have microtubule growing everywhere. So they are growing and eventually shrinking, growing, eventually shrinking, growing, eventually shrinking. So this is a probability to go from plus to minus and from minus to plus. So basically, if I integrate these 4 parameters, I have a velocity of growth, velocity of shrinkage, which can be totally different, right? In effect, they are very different. I have a probability to go from one regime to the other, or to go from the other regime to the other. And with these types of metrics, in principle, I capture totally the phenomenon. And there is no reason for which I'm missing something. And this is one part of the theoretical development of collective effect that is essential. You need to identify the parameters that are going to be key. So far, so good? Yeah? So then the next step is to compute the probability to have, as a function of time, the distribution of states, of polarity. So this is written here, polymer tips. So if the tips is in the plus state, it's going to be equal to minus F plus minus P plus F minus plus P minus V plus dz P plus dt P minus equal plus F plus minus P plus minus F minus plus P minus plus V minus dz P minus. dz is the derivative over the z direction of the plus ends. So again, for those of you who do not do math every day, it must be a bit impressing, but it's not, basically. I will try to get a sense of what each of the term means. This is the variation of the function of time of the plus, right? So you want to count, actually, what is coming and what is disappearing. It's like, you know, the metaphor of Feynman yesterday, right? What comes in your account, what goes out. Exactly what you do. So basically here, you count the ones that were basically in the plus state, which are going to leave the plus state to commute to the minus state. So you lose them. So there is a given density here, multiply the probability to go from the plus to the minus, and since you lose them, it's a minus. Because it's like, again, you go to the movie theater, you take a bank account, you have less money on your bank account. Here it's a plus.
What is rho?
What is the P plus?
Rho, it's P.
It's a P. P is the plus tip, it's a distribution of the plus ends. Either you are in the growing or in the shrinking phase. These are the two states which are existing. If I have an end, is it in the growth or the shrinking phase? So you want to know on average, are they growing or are they shrinking? Okay? So this has left the plus state and it went to the minus state, so you lost it. Here you're going to gain it. It was in the minus state, P minus, and it's getting transformed from the minus state to the plus state. So it's a gain. You lose, you gain. The last term is probably the most complicated one. It's basically the gradient over the z-axis of the plus. Basically, there can be sometime more or less plus, and there's going to be a gradient associated with the velocity of growth. Depending on the sign of this gradient, you will lose or gain. Basically, it's any difference in concentration. Somebody yesterday was talking about gradient of particle. I don't know who it was yesterday or the day before. If we imagine there is a gradient of the plus, you need to take into account also in terms of loss or gain. Okay? I'm doing the same reasoning now for the minus population. The variation of the function of time of the minus population, I'm taking the one that were in the plus state, that eventually going to commute to the minus state, so it's a gain, minus the one that were in the minus state that are going to commute to the plus state, plus the gradient. And again, depending on the sign of the gradient, I will have different results. This is a traditional, out of equilibrium set of equations. For those of you who made statistical mechanics, it's essentially the master equation, basically. You take you have two populations, the growing microtubule, the shrinking microtubule, and you want to evaluate the loss and the gain. There is nothing mysterious here. It's really like there is no disappearance of matter, basically, and you want to know where things go. Where do they come from and where do they go? These type of sets of equations are very generic. You will find all across the board for self-organization processes out of equilibrium. This is really the core of, it's an example among hundreds, thousands of examples of that type. Is it okay? The more you ask questions here, the better, right? Yes?
In the first equation, the third term,
Yes.
should we not consider the velocity of depolymerization which should be associated with the loss of the plus states?
Yes. Basically, you're saying that how come for this state here, we care essentially about the plus population. This is a very good one. I will make a tentative answer, and then I need to think more whether what I'm saying makes sense or not. Is that here, what you care about is gradients of the plus. So since you care essentially about gradient of the plus, they are inevitably associated with the plus velocity. So somehow it is this population that you care about. You compensate for the gradients of essentially this one. These ones are less contributing to this term. I think your point is that you would have expected this term to be associated with. This is the point that you are making, to which I'm trying to answer tentatively. I get your point. I'm trying tentatively to think about the fact that if you wish, here it's symmetrical because you loss and you gain. Here it is not because it is the gradient that you care about. We can discuss more. I'm 80% sure of what I'm saying, but 20 doubt. So we can discuss more. Other questions? Yes?
Is it only to when the ratio and the catastrophe are well separated? So, you're saying that the other part doesn't, B minus part,
Yes.
doesn't contribute to your first equation because it is very negligible as compared to the B plus part.
This is a negligible argument, that somehow one is going to contribute much more.
They are well separate, they're at distance. have both the process happening at your
Yes. So basically, all this starts to work if you have an interdistance between them, which is sufficiently large so that they do not contribute to the same extent. That's the point that you are making. So I'm in a regime where essentially they are sufficiently far apart so that there is no internal contribution. This is, I think, the point that you are making, that somehow it's a density argument. Yes?
If we have time, can you please again define these terms again? F
Yes.
plus minus, F minus plus,
Yes.
F minus.
So V is the velocity of growth. V minus is the velocity of shrinkage. So basically it corresponds to the rescue and catastrophe, right? So basically it's what is my velocity when I grow, what is my velocity when I shrink, okay? This one is the transition between states. So you see here, if I was in the plus state, it's going to be the transition to go from plus to minus. And I have a minus here because it's a loss. Okay?
If you have a minus here, that means the entire term indicates loss
It-
polymerization.
Yes, right. So
Or just the initial term.
it,
The
it,
entire long.
it means that somehow if I have a distribution of,
Yes, got it.
you got it?
Yeah.
So basically it's, this is the loss, this is the gain,
Yeah.
taken throughout all my structure. It's okay? And here it's the reciprocal because here I care about the P minus.
It seems like if we bring this last term in the left hand side and we take common this P plus term, it's kind of the material derivative, like you take the jump,
Yes.
but along with this, somehow it's flowing with this speed, like it's like has speed, so it's like changing the P plus, but along the changing this plus, it's also moving. So that's called the material derivative, like
This one here.
Yeah.
This is the term that you're talking
This
about.
term
Yes.
is the first term in the left hand side.
Yes.
So it's can del by del T plus V dot del.
Yes.
That's
Indeed. Yeah. Yeah. Other questions? I think I would like a question from the most biologists in the audience also. Don't be shy. You can ask simple questions. Okay, maybe one parenthesis. Whenever you're going to do some computational, if you use this in your work, maybe you won't use it somehow. You don't need to be a mathematical solver or good theorist to solve the equation. But what you need to do absolutely is to understand the basic equation. Because if you don't go into the basic equation, it's becoming a black box. And then it's impossible to test whether or not what you say makes sense or not. So at least this equation, you need to grasp the meaning of each of the terms. Yes, please.
Yeah. Can you add another term in the first equation, let's say for V plus
Yes.
derivative of P minus?
V plus derivative of P minus, no, because here you are in the plus state, and basically it's the derivative of the plus state that is going to be affected with the velocity that they have. So couple term like this, I don't believe it would make sense, maybe I'm wrong. But here I do think it makes sense, and we had this argument before, or why not we add this term to this term? But the coupling, I would not see why. Because you are growing, right? If you are growing at the given velocity, the derivative of P plus, you contribute to this one and not to the other. One question from the biologist, pure biologist, don't be shy. I don't remember who is biologist in the audience, so just, okay. If he doesn't come after the lecture. Yes?
Sorry.
Yes.
P plus and P minus.
Yes.
The number of strands that are growing and
Yeah.
number of strands.
That's correct, yes. Yeah. Yes? Yes, this one is not easy. This is a gradient. Gradient is a difference in concentration. So basically, when I'm moving along the z-axis, I'm going to have a lot here and much less here, right? So I'm caring about this gradient here, plus the velocity of which that they grow. And whenever there is a gradient, basically there's going to be a compensation between this gradient and the population. This is taking this term into account. Basically, it's taking into account the fact that the variation in concentration over the z-axis is going to contribute to the population of plus. This is the intuition behind it. So here is the transition between states. Here it's incorporating the gradients along the z-axis. Why I'm insisting so much in this equation is that because of this problem of collective effect, you are bound to be going to these equations. Because if you wish, they integrate populations of polarity, transition between states, gradients, and somehow you would find different flavors, but it will always be equations of that type. Whatever problem you think about, yes.
So you're always taking this one-dimensional growth.
Yes.
Negating the xy.
Yes. So it's one-dimensional because it's tractable to some extent analytically. And as soon as you go to 2D and 3D, computer simulation makes the job. But basically, 1D, it's allowing to have intuitive meaning of what's going on. But indeed, and basically, this is what I'm going to show the solution in 1D, but there is a reference that I will give you, many papers around 3D simulation with this constitutive equation, with many molecular motors added, and so on and so forth. But basically, there is no dramatic novelty in 3D. Most of the physics, if you wish, can be captured already in 1D. So, yeah, there's some benefit.
Is the left-hand
One last, and then I move.
Is the left-hand side that
Yes.
it is some unit?
Okay. So it's variation of the function of time, actually, of the plus population. So if you wish, it would be the variation of concentration over time. It's a dc over dt.
If we are equating that,
That's right. So basically,
third term according to it.
what do you say?
Third term.
The third term, well, the third term is going to be, you are still okay in unit because this is, it's okay? Because it's a velocity, so it's a, yeah, they cancel out. So it's a unit okay. So what's going on when you solve these sets of equations? You end up with something funny, which essentially they observe and they report in the paper, is that there's going to be length as a function of time of the microtubule. There are two states that are existing. A state in which the microtubule reach a finite length, which we called bounded, and a state that is like this, that was called unbounded. And this is the result of the numerical simulation. So I'm repeating. If you wish, they have this surface. They let the microtubule grow, shrink, grow, shrink, grow, shrink. And what they observe is that somehow the average length of the microtubule are going to be, depending on the sets of parameters, V plus, V minus, F plus minus, H minus plus, they're going to be either bounded, bounded means limited, that somehow the length will be limited, or unbounded, meaning that they will continue to grow, 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 grow. That's the result of the simulation, and the last part of microtubule is going to be trying to understand what's going on between these two states. Yes?
Those individual fluctuations,
yes.
How do they span out with time?
How do they?
How do they span out with time?
Over time. So this, they haven't said in detail in the paper, as far as I remember, but it has been measured experimentally and probed actually. But this is heavily dependent, as we will see in a second, on the parameter F plus minus, F minus plus, because somehow these fluctuations are going to be key. But one of the key points is that growth and shrinkage are extremely different. Growth is very, very progressive. Shrinkage is, poof, it disappears, which actually motivates the name catastrophe. They named it catastrophe because maybe I'll show the movie tomorrow morning by starting so that you see illustration of what I'm talking about here, but somehow it's a growth, very, very progressive, and then boom, disappears. So yeah, there was another hand, yes?
So how do you get those values from experiments, F plus minus?
Yes. So at the moment, everything is numerical. These are theorists who are thinking out of the box with first principle, this type of ideas, polarities, ensemble of objects, etc. They don't care about experiments. At the moment, they think autonomously, far from experiments.
What values do they get?
They don't get values. That's
some quantity they have to.
This is the point. This is the point that they care about is essentially these four guys. And this is my last question on this part before the last half hour when I will go to one part. actually, is that I would like you to try. First of all, do you see the consequence here? Bounded, right? It means that now I have a wall. I'm growing microtubule everywhere, and when I'm bounded, it means that the microtubule, whatever I do, reach a finite length. Okay? And for another set of regime, when I'm playing with the F plus minus, F minus plus, V plus, V minus, I'm going to another set of regime, and then the microtubule will grow to infinity. So it's a major change, right? It's a total change. It means that I'm going to a state where all the microtubules have a length which is around the cell to a state where they grow. Somehow it's not very different from going from here to here, right? Here I'm finite in length. Here I can start growing very dramatically. And there is no way I can get this by hand wavy. I need to run the math to be able to have this point. Yes.
So you change these parameters and you will come up with different.
That's the point. And the point that I'm trying to ask now is to get a sense of what is this threshold, basically. Is there a guess of how can I go from here to here, basically? How can I go from this regime to that regime? You could guess it. I read it, so I have no merit, basically. But you could guess it by having an intuition, actually. Try to play with this four quantity. Yes?
Sir, so bounded means the rate of polymerization and rate of depolymerization nearly are similar.
Yes, but the point is that I agree with you, but it is more subtle because you have a dynamic process. So it's not only rate of polymerization, depolymerization. It's a combination of velocity of growth, transition state. So, I mean, using one word is satisfactory because it goes in the right direction, but it can be wrong, right? I mean, you need to run the equation. You cannot escape them. It's like a Toner 2 or Vitschek, basically. You need to, I mean, you can ask, you have a practitioner of Vitschek model. You need to run the equation. Otherwise, I mean, correct me if I'm wrong, but intuition can be very misleading. Yeah. So the combination of parameter, we're coming to scaling argument that I was presenting the very first day. In scaling argument, De Gennes in France was using a lot scaling argument. It's basically, you know the parameter, you play with units, and you end up with a result which is not unreasonable, and usually right, actually. So basically, I'm going to write the point, actually.
If you take a different cells.
Yes.
If you take a different cells.
Yes.
So this phenomena will be averaged out in the end.
Yes.
You will have same scenario.
That's right, but for different reasons. That's the whole point. Is that for different reasons, and that's the parameter I'm going to write right now. It's a combination. Basically, everything is going to make a transition whenever you have the product that is here, V minus F plus minus equals V plus F minus plus. And that will answer partially to your point. It's somehow, you go from bounded to unbounded when this is going to not to be true anymore. Let's try to get a sense of what's going on here. Here it is in a shrinkage phase, right? And I'm going from the minus to the plus. Here I am in the growth phase, and I'm going from the plus, and I'm going from the minus to the plus. No, sorry. Yeah, I'm going from the minus to the plus. So basically here, when I'm compensating exactly the probability multiplied by the velocity, it is at the point at which the transition occurs. If you have a hard time viewing it, you have another way to write it, which is V plus or V minus over V plus equal F minus plus over F plus minus. When the ratio of the velocity is equal to the ratio of the transition, basically that is the point at which there's going to be something happening. I'm repeating one last time, stated differently. If basically you exaggerate the transition from the minus to the plus state, you will be essentially transiting to the length as long as you respect this relation, basically. The ratio here, if this ratio is larger than this one, you go to unbounded. If this ratio is smaller than this one, you are to bound. So returning to your point, experimentally, it would mean that you have a cell, you measure V minus, V plus, F minus plus, F plus minus, and you check in which part of the regime you are, depending on this equality. If you are lower, you are in one regime. If you are larger, you are in the other regime. But it's not intuitive because it's a combination of 4 parameters. So you need to go through the measurement of the 4.
So how difficult it is to measure experimentally?
Okay, this is work, but not difficult. I will show them rather tomorrow the images of live cells, but you can image microtubule in single cell, and you would see, maybe I'll start by this tomorrow morning before the lecture on systems biology. You will see the growth, shrinkage, so it's measured, but nowadays.
So what will be the use of this model?
Yes.
If we come up with the right model,
yes.
How much implications can we also find out whether some disease cell
yes.
Which are doing something different from the normal cells?
That's a way to go. Yes, the whole point is really this one, is to be able to say, well, for a cell to be more polarized, they would need to be, let's say, going
cells
to the...
have different behavior.
That's the whole point, exactly. So either you're going to play in the complex of proteins that are going to ask to add, to work on this part, on that part, or this part, on that part. And then you probably will be able to transform this guy into that guy. To make it more concrete, one of the defects of cancer cell, but this is a very hand-wavy argument, is to be overpolarized. So if you want to correct this overpolarity, you have to have a molecular actor. But in addition to that, you need to go to a regime where eventually you're going to be bounded, basically. So basically it means that you need to look for this condition to eventually revert back.
This can actually tell you whether there's something wrong with this.
Absolutely. But the point I'm trying to convey here is that the molecular action is key, and this is headed by chemistry, for sure. Chemistry is the key element here. But the ensemble phenomenon is a result of such computation. Because it's too complex to do it with the brain. It's a combination of parameters. Yes.
My question is related to
yes.
someone's question.
Yeah.
So, why don't we see a third state where it simply falls off?
Falls off?
So
there could be a state where eventually it disappears. Yes, yes. So, because what I put below the carpet that I haven't shown actually is that there is a nucleation rate. So basically, it keeps nucleating. So basically, disappearance would happen if there would never be nucleation here, but basically, we are always in a state where it keeps nucleating. But I totally agree with you, it would totally exist. It's okay? As far as I remember in the paper, no. That's the whole beauty of the model, is that either you are bound and limited, or you go from one state to another. It's a typical transition, right? It's that you go from one bounded state to an unbounded state. And the richness of this dynamic system is really this one, is that you transit from one phase to another phase, and for the non-physicists in the audience, the dream of a physicist is to have a phase diagram where you have one axis, another axis, and you travel and you know where you are. This is the paradigm, if you wish.
It tells about the final state,
That's right.
not the kinetics problem.
It can tell both, with due care. Because you can have some hysteresis, there can be all kind of... Basically, the punchline of all this is that the more I've been learning or interacting with colleagues actually in this matter, is that you need to run the calculation all the way through before you can say something. It's otherwise very misleading. So let me summarize, tell you one more step that would be important for this afternoon, and I'll say one word about actin. That was the... Tomorrow I would like to move to systems biology. So basically I would like to say one word about actin. I need to, I'm summarizing where we stand, and then I present for this afternoon that something that we will need to explain the motion of our pond water, so organism. So I started by the problem, right? Chirality was present yesterday. And then the point that I was trying to say is that if you go to single biopolymer molecular motor to an ensemble, which is the majority of the case what's happening inside the cell, you are bound to be studying collective effects. This collective effect, because of this problem which starts already at 2 or 3, requires math, requires equation. And then I was going through the description of some elements of the cytoskeleton. microtubule and actin filaments. I was emphasizing this morning essentially on microtubule, just by arbitrary choice. I could have started by actin filament, but basically microtubule were the simplest one to start with. Then I was saying that cytoskeleton is a term that is not very correct, in the sense that it imposes cell shape or contributes to impose cell shape, but it has an intrinsic dynamic which makes the system has some kinetic, and the kinetic is key to eventually understand cell shape and cell motility. I was going through the GDP hydrolysis cycle of tubulin dimer and protofilament, giving after that the dynamics of the microtubule, which are growing at a velocity v, and then shrinking, growing, shrinking, growing, shrinking, and self-organized around a structure that is a centrosome, which allows the cell to find its center. Eventually, when there is dysregulation of this microtubule dynamics, it can polarize a cell, which gives this structure. And the last point was the way of saying that I was hand-waving my argument, but then you need to go to math because you integrate hundreds of objects interacting in concert, collective effects, self-organization, and the key element is now to find the proper parameter to make the model. This was done by one PRL, Stanislas Lieber and Marilyn Doctorum. It is one example, again, of thousands, but it has the merit, I think, of being quite comparable with what I was describing before. They needed to identify 4 parameters: velocity of shrinkage, velocity of growth, transition between plus-minus state, minus-plus state. And this was essentially master equation counting objects. If I have a variation of sign of plus, it's what disappears, what comes, and compensation for the gradient. And the same for the other state. And after that, it's hard work to solve the equation. And eventually, a very simple result, bounding growth or unbounding growth, with consequence for polarized cell or non-polarized cells. It's okay? Now, for this afternoon, we will need something else about microtubule, and for this I'm going to show you schemes, because the swimming organism that you saw yesterday, that you started to describe with your gesture, so it was this, or it was like this, which I will have done the same, so basically it's basically what it has. The basis of this are flagella and cilia, and try to guess what is the core element of cilia and flagella. It is microtubule, right? So that's why I was deciding to go through microtubule this morning, because the key actors allowing your microorganism to swim this afternoon are microtubule. And I need to introduce you how flagella are made of and what cilia are made from. And for this, I will turn to this to show you all this. Again, it's molecular biology of the cell, so it's a classic. So you will not see it under the microscope, but I'll try to show you 1 or 2 movies at the beginning of this afternoon so that you see it with better resolution. But basically, you will see that almost all the microorganisms that you saw yesterday under the microscope, they have some very, very tiny flagella or tiny cilia. We have them also in our body. They allow the mucus to be transported. Basically, these are active processes. The main actors are microtubule and dynein, the motor, but assembled into a structure like this. Extremely, extremely reproducible. This is the structure. Arrangement of microtubule in flagellum and cilia. So this is an electron microscopy pictures. What you see coming towards you are microtubule. The same one as I keep talking about since 2 hours, right? So you have this arrangement of microtubule in flagella and cilia. And you see here the representation. The resolution is not very good, but you see in green here, these are microtubule. You have dynein arm, which are represented here in red, actually. And you have a system of 9 plus 2 microtubule. The 9 microtubules are all around like this. And in the center, you have the 2 microtubule. And in between, essentially, you have interaction between these microtubule and the neighboring dynein arm. Try to guess what's happening. If you have the dynein arm, which are here interacting with their neighbors inside the rod. Yes, you make the gesture. But somehow imagine you have many rods like this. You have a molecular motor action like this. If you do like this, at some point there's going to be an imbalance, and you start to see that there's going to be a bend, actually, that is going to happen. It's okay? You need to feel it. It won't give you orders of magnitude, but at least, whenever you can try with your neighbor during lunchtime, right? You take your neighbor and you try to apply a force like this, and you try to organize it to 9 + 2 in a rod, you will bend. Yes. Yeah. There's going to be something like this. You feel that there is going to be a broken symmetry. It's going to go in one direction because it's perfectly organized here. This is an EM structure, but the image is beautiful, actually. You see here, it's perfectly organized 9 + 2. There is an interaction here that is extremely robust and nicely organized. And you should imagine that it is really going in this direction. So it's a flagella or cilia that are going to be quite long, actually. So this is not an imagination, because if you take a look at a real movie, and here it's an example of a spermatozoid actually dynamic, where you see here the motion actually of the spermatozoid over time, where essentially it looks like this. You have a state, you have a contraction of the dynein interacting with the microtubule. This is going to give a bend in this direction, and then there is a recovery stroke which is moving in the opposite direction, and then you return to the cycle. So the bend that you were mentioning a second ago, that's basically the bend you would see in the gliding of a spermatozoid. Why am I introducing it now? You will see this afternoon in the pictures you took yesterday that all your structure were having, not visible with the resolution that we had yesterday, but that's the core element of their swimming. It's microtubular organization with dynein constituting the main element that they have usually in their surrounding, allowing them either to rotate or allowing them to go straight. And you will see that this afternoon, essentially. So basically the point I'm trying to convey is that everything we did until now for microtubule and centrosome, you could imagine that you could repeat it, but for this problem. It would be microtubule organized 2 + 9 plus molecular motor with potentially some dynamics of growth and shrinkage, and then you return to a set of equations with prediction. It's okay? What is amazing to me, actually, is that the very same structure here that was in structure like this is so much concerned across the cell matter and living systems, right? You find them in organisms of this afternoon. Again, we find them in our body. You find them in the spermatozoid. You find them all across the board with this 9 + 2 plus dynein motor. It is very, very highly concerned with the same function, which is basically to have this type of motion. One last element before the 10 minutes of actin. You were showing this bent, and you were doing this also. Who was doing it? Yes, why were you doing this? Okay, you were doing something like this. It will be important for maybe this afternoon or Friday. When you see this motion, what impression do you get when you see the movie of the spermatozoid moving? Yes? Is it chiral or not? Is it chiral or not? It seems, is it? Okay, I'll let you think about it. Does it have a broken symmetry or not?
Yes.
Is it indispensable?
Expensive, you said.
Indispensable.
Indispensable.
Asymmetry, directionality.
So think about it, right? Basically, there seems to be the broken symmetry at the single filament that was yesterday. The whole point of today's lecture was going to the ensemble and to try to see if the ensemble is having also a broken symmetry. So let's, at the moment, pause on this one, and we'll return to that on trial. Okay? The last 10 minutes, I'm going to do a little bit of acting, but it will be very short, just that you have a glimpse, for those of you who never saw it. So by the same way I was introducing microtubule dynamics, I'm going to say one word, and it would deserve 2 hours at least, about actin dynamics. So if you take a look at an actin filament, it's going to have, as you remember, it's a double helix, but I'm going to make the picture a little bit easier. It looks like this. Two actin filaments which are intertwined, actually in this manner, in a double helix. And it has one end which was conventionally called plus, which is the fast growing end. And one end which is called minus, which is called the slowly growing end. You won't be surprised to hear that there is in the medium ATP bound to actin monomer. Here, sorry, it's going to be ADP that I need. And here it's going to be monomer of actin bound to ATP. I have a given probability to bind here and a given probability to detach the monomer associated to ATP with a K on over K off of about 10. Here, I have a given probability to bind and a given probability to unbind of monomeric actin complex to ADP of a K on over K off, which is about 0.3. So it's a way to say that if I take an actin filament, again, it's intrinsically a dynamic structure. I cannot say I have an actin filament, it will be the same length forever. It's wrong, basically. Every minute, it's changing its length, and this is true throughout the body. So it's a very dynamic system, turning to the same argument as before. It is dynamic because every actin monomer can hydrolyze, this time it is ATP, whereas microtubulin could hydrolyze GTP. So it's not the same source of energy, but essentially the idea is very conserved. I'm going to do it with my hand. You see that K on over K off of monomeric ATP actin is 10. So basically there is a high probability to bind and a low probability to unbind. And here there is a high probability to unbind versus a low probability to bind. So basically I'm making asymmetric the two ends. I have one end here which is going to grow and another end that is shrinking rather than growing. So I have something like this. The name that was given to it is treadmilling. So I don't know if you're familiar with the word treadmilling, but somehow it really, you see what it means, treadmilling? In French, it doesn't mean man, so that's why, but if you have, that's right. It's something like this. You see that it's continuing doing like this. So all this reasoning, I'm limiting it on purpose to a single filament. So if you wish to make a parallel between what I did, actually, I'm essentially repeating this step that was here. I'm trying to repeat it one more time here. This was the single microtubule dynamics. Here is the single filament dynamic. But it's very different. Here it's a rescue, catastrophe, rescue, catastrophe, rescue. Here it's a growth, depolymerization, growth, depolymerization. It's a very smoother, it's much smoother dynamic. You probably guess what I'm going to try to do next. This is a single filament dynamic, but in real life, I don't have one. I have thousands. I have 10,000s. Basically, I have an ensemble of filaments interacting in concert. So come back the problem, how do I go from a single filament of actin to an ensemble of filament of actin? So I'm giving you one example of many. As you know, actin is present all across the board in the cortex of cells. It's called embryo, it's called cells, it's called tissue, and so on. I'll give you one example because it's a quick one and easy to show. Whenever you have a cell that is moving on a surface, it's forming at the front the so-called lamellipodia. This structure, this is a cell, this structure is made essentially of actin. If I'm depicting what's happening here at the front, and I'm going to zoom dramatically, what I have here is that I have nucleation of actin, which is growing and forming a branch network of that type. Each of these filaments here correspond to a single filament over there. So basically here, I'm forming a very, very connected network of actin filaments which are polymerizing, depolymerizing, polymerizing, depolymerizing. So the term that was given to this was a term of active gel. For those of you who are chemists in the room, you must have been working with polymer, right? So imagine you have polymer, reticulant, and you have a polymer that is out of equilibrium so that it can grow. So imagine you grow, you cross-link, you grow, you cross-link, and then if you have 10 to the 4 filament connected, you're going to have a gel. But the gel is going to be a tricky one because you have some parts which are growing, some parts which are not growing, and you feel what's happening if you have part of the gel growing and part of the gel not growing? Yes, I heard it. Yeah, space, yes.
That become spatially deform. It would deform, but not.
Yes, you feel that there is a problem. If you grow somewhere because you have this growth process and somewhere else you don't grow, you have a stress generated somewhere, and then you come to a problem is how should you write this constitutive equation to eventually predict where is the stress is going to be applied compared to which place. So basically, everything that I said here can be revisited with another set of equations, other formalism, other experimental tests, but the spirit is the very same, is that you have the basic constitutive mechanism for the molecular scale, and you need to translate it to an active gel, and then you are back to self-organization. Because here, we go from 7 nanometer in scale to a structure that is about 5 micrometer in scale. So again, we are going to a factor of 10 to the 3 larger scale, for which hand wavy is not anymore good, and you need to run equation and test prediction of models. So it is the last point I wanted to say somehow. One last time. Collective effect is at the core of cell motility, self-organization of tissues in developmental biology. All across the board at the moment, it's an international vision that is starting to converge, actually. It requires equations. You cannot bypass them. It requires also a deep knowledge of the single filament behavior. But the integration from one to the other requires all the steps that I said. Microtubule dynamic instability, integration through the master equation, dynamics of actin filament integrated to any type of structure, and the equations associated to it. Time for the last questions before lunch. Yes?
So overall what I could make is, having learned all these things until now, so it seems like when the microorganisms move, so they don't decide where to move, they just, for example, in a local area where stress is created, because of these systems that are playing inside them, the actin and everything, so they sense it somehow, and then they're forced to move in that direction. They did not decide it.
A microorganism has no free will. It doesn't decide much.
That's what I'm saying.
Yes.
So their entire life depends on these forces around them. Is it so?
It's a good start,
Yes.
but it's not the end of the story. Basically, you will see this afternoon, and actually, I'm hesitant for the Friday paper I want to present. I'm hesitating between 3 papers. One of the papers is going to ask your question. Whenever I have a bacteria moving somewhere, why is it starting to move straight at some point, and why is it going to stabilize in a place where it has food and can reproduce? That's the question you're asking. It's somehow how a microorganism in pond water is going to go in a given spot because it's better for that's what you're saying somehow.
Yes.
What informs the microorganism that it's good? So I haven't answered to the question yet.
No problem, sir. Means that it seems like that physical principles are defining life.
They're playing, together with chemistry and biology, they are contributing, yes. This is the core message, that somehow you need to integrate this, but the last part that you are asking is why is it going to stabilize in a place where, because if your systems swim all over, they will never stay in the same spot. Think about, there was one that was moving like this. If it's doing like this all the time, it will go to the other end of the picture taken by Pramod, and it won't be in the right spot. So, you need to invent.
Physical consequences could be, physical phenomena could be consequence of chemical.
I totally agree. It could be a consequence, it could be the cause, and both exist. Indeed. Shall we continue after lunch? Okay? Yeah, sure, please.
When you are like this cross-linking,
Yes.
over cross-linking can lead from gel
Yes.
to a precipitation.
Yes. So it can give actually it can be a way actually to get rid of the structure. Because at some point, you need the lamellipodia to disappear. And so somehow precipitation, which would be essentially collapse actually of the structure, would be beneficial because that's the best way of getting rid of it. It's not yet degradation of the protein, but that's a way to, to suppress the lamellipodia so that it's going to grow somewhere else. So both growth and disappearance process could have a functional role. Yes.
This liquid phase and this jelly phase are in equilibrium, of course, because of this kinetics.
Yes. So the both of them are out of equilibrium. In essence, they're out of equilibrium, but the phase diagram, or basically I would say differently, the state that allowing them to go from solid to liquid is one that is extremely difficult to define in a univocal manner. Because you see, if you are solid, you can deform, you can apply stress. As soon as you become liquid, this is going to disappear, but depending on the phases on which the lamellipodium is, you can transit from one to the other. But again, you need to rerun the math or the physics to eventually find this point and then transit. But yeah. But I think you would find most of the reactions that are in the chemistry laboratory, but here they are self-organized by the cells, which is tuning these parameters. So shall we have lunch? Okay, see you later.